artists, Professor Lutzel, who is at the Scripps Institute. He actually does scientific art, and this is not computer generated art. This is a drawing made with paints. He actually goes through all the protein structures, decides how to paint them, eventually builds up the painting. And he was very nice when I asked him, contacted him. Within an hour, he not only gave me permission, he sent me the final high resolution image and says, please use it. So we would like to thank him for giving us permission to use this thing as a poster. And with this, it's time to then switch and come to today's session. And it is a pleasure to introduce Anusha Singh. Anusha is a biology student at the IFR. She is from Kolkata and has landed up here working on something to do with the epidermis of zebrafish, which is her research. But that's not something she's going to talk about. She is interested in all things that go viral, including the bugs. And today is going to be about things that go viral. So Anusha, all yours. And just remember that you have to shout for the audience. The mic is only for the webcasts.
Okay.
And if you're following us online, if you want to ask questions, just type it in the chat.
Am I audible to everyone? Great. So hi, I am Anusha and let's just jump right in. So we have heard a lot of things about coronavirus and what is the epidemiological pattern of it and what is the virulence and what is the fatality rate, but we are going to start with something very simple. So we are going to start with a very simple question. What is disease? So can anybody define disease for me?
Something that puts you at unease.
Sorry?
Something that puts you at unease.
Something that puts you at unease. Good. So basically WHO, World Health Organization, has defined health as a state of physical, mental and social well-being. And disease is any condition in which this well-being is perturbed. So this well-being is negatively impacted. So this is disease, which is absence of health. So then disease can be of two types. It can either be communicable, like chicken pox, like it can go from one person to another by touch, or it can be non-communicable, like diabetes, which cannot travel from one person to another. So then we'll move on to the next question, and that is what makes you sick. So what are pathogens? Pathogens are anything that makes you sick, anything from viruses to worms. So this is a huge range of things that can make you sick. And what they do is basically they affect your body's normal functioning and cause your body to behave in a way that is not normal, that is aberrant from the normal condition. So how do we define pathogens? We define pathogens using three different criteria. The first thing is it has to use your nutrients for its survival. That means it has to use your resources, your body's food, your body's food to make its own food. And so there are pathogens which can also steal your food literally, but most pathogens use your resources. Then there is anything that can be communicated, that anything that can be transferred from one person to another. And the third is anything that negatively affects your physiology. As I said, anything that harms your body and makes your body act in a way that is not normal. So then what's the next question? The next question is, what is a virus? So viruses are really cool creatures and they are at the borderline of living and non-living. So as of yet it is still a scientific debate question whether viruses are living or non-living, because viruses can only survive when they are infecting a cell. Otherwise they are not considered alive because they cannot reproduce. So reproduction is the basic biomarker of being alive, and if something cannot reproduce, it is not considered to be alive. So viruses are only considered to be alive when they are infecting in a cell, and that is why we call it an obligate pathogen. That is, it is obligated to infect something. So this is what a virus basically looks like. Virus is just something which is genetic information coded in the form of DNA or RNA, which is encapsulated by certain proteins. This is the basic idea of how a virus looks. You have your RNA, DNA over here and your protein coat over here. Yeah. So this is the basic structure of a virus and this is what it looks like. So viral diversity. Now we will go on to what viruses can be like. So this is a very interesting virus. This is the first virus that was used as a vaccine for smallpox. Then this is something you've all had: influenza, rhinovirus, common cold, respiratory viruses. This is something we've all had. And this is a very special bacteria, virus, which infects bacteria. This is called a bacteriophage. And this is the famous Ebola virus. Yeah. And so this is the size. So this is the size bar. So this says 100 nanometers. So if you roughly see this, HIV is around the size of this size bar. So it's around 100 nanometers. So then, but what exactly does 100 nanometers mean? So I'll give you an idea. To give you an idea, your hair, the thickness of your hair is 100 microns, which basically means that this virus is 1000 times smaller than your hair. So what makes viruses travel? So viruses can travel by these different modes. So the first is direct contact, which is through between the infected person and the healthy person, which is an example of which is chicken pox. Then there is droplet contact. When you cough or sneeze, viruses are loaded into these respiratory droplets.
You can stand here, look at us and use the clicker. You have a clicker.
Oh.
It's like, is it on?
It's on. It's on, but it's not working.
You have not connected that. You have not connected it.
Huh.
So now you have
Yeah. So great. Thanks. So yeah, where were we? Yeah, droplet contact. So the next way by which viruses can travel from an infected person to a healthy person is by air, which is through droplet contact. And the disease that is spread this way is called the common cold. Then there is indirect contact, where the virus can stay on inanimate objects which have been touched by the infected person, and then they are later touched by a healthy person and transferred to the body of the healthy person, where they can cause infection, which is an example of which is measles. Then there is the fecal oral route, where the infected person sheds the virus particles in their feces, and which may contaminate food and drink and then travel to a healthy person. The next, and polio is a good example of such a disease. The next is vector-borne diseases, which are transferred from an infected person to a healthy person by means of a animal vector in the middle, which takes a blood. So like for an example, like mosquito takes a blood meal from an infected person with dengue and then transferred it to the body of a healthy person. And the last is animal to human transition. Now this is a little different from vector-borne diseases, because here the animal also suffers the disease. Here the mosquito does not suffer dengue, but here for example, if rabies is transferred from a rabid dog to a healthy person, then the dog is also suffering from rabies. So now we will see a video of how droplet transmission happens. Yeah, so what happens basically is that viruses are encapsulated into a water droplet which you sneeze out, and then they float through the air, and then they can reach an uninfected person and then can cause infection in that person. So why do we need to study these viruses? But not just because I mean for me they are really cool, so that's why I want to study them, but why would the general public want to know about viruses? Why would you want to care? The reason is, you would want to understand the pathophysiology. The pathophysiology is any way the disease makes you sick. So you have to understand how the disease is making you sick, so that you can choose which place to target. Then you can, so this is the drug developmental process, and this is, so you first select your target, then you choose how to target it, then you do some chemistry a bit of chemistry here, and then you do in vitro and in vivo studies to test out the drugs in the animals, and then you do clinical trials whereby you test it in humans. So to do all of this, you need to understand the pathophysiology and how the disease makes you sick. So that is why we need to study the viruses. And the second reason is to understand transmission. So I will come back to this, but what I want to say here is that diseases can be transmitted from one person to another like this, like a chain of events, and we have to understand how this transmission is happening so as to be able to control it. Otherwise, as in the case of the novel coronavirus, it has been spreading from one person to another, but we don't really know how it is spreading right now. So that is why we need to understand this so we can control it better. So I'll come back to it. I'll explain it to you. Yeah. If anybody has any other questions, please stop me in the middle. So what is epidemiology? So this is epidemiology is the science of studying disease, how diseases spread. So diseases can spread from from sick people to healthy people, and this when what we study is upon study people study. So study of people who are sick. This is what epidemiology is all about. And in epidemiology, we study three things. We study what the host is, the host factor, what the age of the host is, how the host lives, etc., etc., to understand how the host can get a disease. We study the agent, that is the agent that is causing the disease, as to how it can cause the disease. And then we study the environment in which the host and the agent can interact with each other to cause the disease. So as I told you, I will come back to this. So this is R naught. R naught is basically the reproduction rate of the virus. So it is a measure of how quickly the virus can spread from one person to another. So for example, for hepatitis C, which is a virus which is transferred from the blood of one person to another, so it is generally via blood transfusions. So this R naught is very low, so 2, because not everybody keeps going around giving blood transfusions every day. So the probability of a blood transfusion transferring hepatitis C virus is much lower. However, when you have something like measles, which is transferred through respiratory droplets and indirect contact, it can infect a lot of people. So one infected person can infect 18 others with measles. So that is why the R naught of measles is 18. So for those Ebola, the R naught was 2, and for SARS the R naught was 4. So that means SARS is twice as more infective as Ebola.
How do you calculate or identify?
Yeah, exactly. So this is how you calculate these. So this is based on the infectious period. So how long does a person remain infective? So if I get a viral infection at a day 1, and then I am infective till day 10. So I am infective for 10 days. So that will increase the amount of chances of infecting other people. Then you have contact rate. So what is the rate of contact? So if, just as I said, in case of hepatitis C, when it's through blood transfusions, it's not, the probability is not very high, but through measles the probability is very high, because you might cough and sneeze and then you might send on the infection to another, and that is the mode of transmission. And the contact rate is the density of people. So it matters as to where you are living. So if you are in a very less dense population, then you won't have the chances of interacting with people. So even if you are suffering from measles, but you are in America, let's say, then you are far away from another person, you won't have the problem. However, if you are in Mumbai and crowded Mumbai, you really don't have the option to sneeze in a very hygienic manner, so then you will infect other people. So this is to explain what the importance of R naught is. So if your virus infects on an average 1.3 people, then you can see that after 5 cycles, 45 people get infected. However, explain. So if you have a virus which has a R naught of 2.6, then after 5 cycles, as you will see, it will infect this same virus could infect 368 people. So this happens in a chain reaction format. So it's all well to know ki
Sorry, why is it so fast?
sorry.
Why is the progression of disease infection so fast? It's just double, right? R naught of 1.3 and R naught of 2.6.
So the reason is because see, so there are 5 people here. Now this, okay, why is this not playing? Okay, so anyway, it's not playing. But so what happens is each of these 5 people will infect one another person. Then you will have 10 other people. So that you will have 5 plus 5, 10. So then you will have 10. So you have 10 people who will infect 1.3 other people. So that keeps on increasing in a geometrical progression. That's why it's so drastic, because you see that the difference in R naughts is not that much, because it's a geometric progression, it travels much faster, and so far after 5 cycles you have a greater disease progression. Okay, so we talk about how a disease spreads and all of this, but it's very important to quantify this, to measure what is going on. So how will we tell if a disease is more deadly or less deadly? We tell this by looking at 2 parameters of a disease, the infectivity of the disease and the virulence of the disease. So the infectivity is a measure of the spread, while virulence is a measure of the fatality, that is how many deaths it will cause. So infectivity means how quickly it can spread from one person to another, while virulence means how many people will it kill. So if it infects an X number of people, how many people in that X number of people will die. So this is a graph to show you how we quantify these diseases. So as you can see, chicken pox spreads very fast, very high rate of contact, very high rate of transmission. So it's very far on this scale. However, something like Ebola doesn't spread so quickly, but is very deadly. So this is a log scale. So you might want to see that this swine flu or seasonal flu, for example, is 100 times more, 100 times less infective than Spanish flu or SARS. And Ebola is 500 times more 500 times more deadly than seasonal flu. So this is on log scale. So the differences are like that. And yeah. Any questions? And so the new coronavirus is estimated to be somewhere here. We don't know right now where it is, but it seems to be somewhere around here. So where do new viruses come from? So we've all heard about novel coronavirus, but what is?
Already 1,000 people have been dead because of coronavirus. Still, why haven't we able to spot it on the graph?
Yeah. So the reason why it's still here and not here is because 60,000 people as of right, I just checked the stats, and 60,000 people have been affected. So the fatality rate matters. It doesn't matter as to 30,000 people have died or 1,000 people have died. Out of the infected, how many of them have died? So out of the 60,000 infected, 1,000 have died. So that is why the fatality rate is not very high. But it cannot be said for certain until the epidemic is over as to what is the fatality rate. Yeah, it can, but I really don't think, because right now it's sort of stabilizing, the numbers are stabilizing. Yeah, so yeah, exactly. So that's a very good point, that this measure of spreading faster can also depend on what precautions we take. Now, if everybody takes precautions against any viral infection that is spreading, then the infectivity of that virus will decrease eventually. So this, if you put in a lot of travel restrictions and etc., then you will have this number decrease a little bit. So yeah, so where do new viruses come from? So where does this new novel coronavirus, where did it come from? So generally what happens is, it's not a new virus as in something that has never been seen in the biological system. So researchers have known coronaviruses for decades, and how they have known it is because all these coronavirus strains have been present in different animals. They have just not been infecting humans, but they are not new viruses as such. They are just new to humans. So what happens here is that viruses can sometimes jump from species, like different species, and come to humans, and this event is called a spillover event, where a virus which used to infect a particular species can now infect humans, and these diseases are called zoonotic diseases. So this is an example showing, so you must have heard of Nipah virus, which came from bats, avian influenza, which came from birds, Ebola, which came from chimpanzees, SARS, which came from civets, etc. So where did the SARS coronavirus hop from? So we don't know right now as of yet, but it is speculated that the Hunan market, which had lots of species of animals on display for selling and everything. So it could have traveled from any one of these animals. It's not right as of now. It's not yet known, because now this market has been disinfected and everything has been shut down. So now we can't really trace back to what animal this virus came from. But it's jumped from one of those species to humans, and then from there on it started from, so the first event, the spillover event, is from an animal to a human, and from then on the transmission happens from a human to another human. So now we'll come to the main part. What are coronaviruses really? So they are RNA viruses. So I remember I told you that viruses can have genetic information in the form of DNA or RNA. So coronaviruses have their information coded as RNA, and what that means basically is that coronaviruses can mutate faster. In biology we call mutations as change in the genetic information of the bacteria, of the virus, and RNA viruses can mutate faster. And there are 4 endemic strains of coronavirus. So you guys have, so if anybody here has had a cold previously, it could have been one of these coronaviruses, this 2293 E alpha and OC43 beta, because these are the most common forms of coronavirus which cause common cold and flu. So it can cause diseases from common cold to something very dangerous like SARS. So this is how coronaviruses look on transmission electron microscope. So we use this technique called transmission electron microscopes to visualize something that is very small. Like I told you, viruses are in the scale of nanometers, so you can't really see them like that. So you need to use transmission electron microscope, and that is why this is the reason why they are called coronaviruses, because they have this halo of proteins around them which surrounds them like a crown. So corona means crown. So I would like to point out a couple of things here. So this is the crown protein, the spike glycoprotein, and I would like you guys to remember this because I'll come back to this later. And then here is the RNA information that is encoded. And yeah, so let's see how a virus enters the cell. So any virus which infects a cell enters it using the proteins it has on its outside, and then it binds to some proteins on the surface of the cell. So this is how a virus is taken inside in the cell. So how does coronavirus enter your cells? So this is very early research work which shows that, so as you can see, so this is the nCoV S protein, which is the spike glycoproteins I told you about, which is present in coronavirus, that and ACE2. So ACE2 is a protein which is found in our cells in lungs. So that is why coronavirus infects the lungs, because ACE2 is present in the lungs, and the S protein of coronavirus binds to ACE2. So as you can see here, wherever there is green, there is also red. I want you to look at these two images carefully, O and P. So wherever there is green, there is also red. So that shows that the S protein binds to ACE2 and thus enters our cells. So what is a strain of a virus? Very good question. So viruses, so we have generally species of animals, right? So viruses, it's very hard to calculate, to divide them into different species. So what we do is we divide viruses into different strains. So there are some strains which are, basically what is different in them is their genetic information is slightly different. So they produce slightly different proteins, so their virulence will be different, their infectivity will be different, so that is, the parameters will differ based on the different strains. And when, so if there are two viruses, for example, smallpox and say coronavirus, then there is a lot of difference between these two viruses. So these are termed as two specifically different viruses. Okay, so that is how they are classified.
How many different types of receptors do you have on your cells?
So that is a very broad question. So because every cell has its own, type of receptors, plus there are common receptors which are present in every type of cell. So you can't really say ki like there is a certain number of receptors which order of magnitude. I would say it's somewhere around thousands, in more than thousands. Yeah. That's true, that's true.
I'm just asking because I'm thinking of the receptor as like sort of a gateway key.
Yeah, 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 exactly, that's how it works.
So how many?
Yeah.
Oh, fair.
Exactly. So then this is what happens exactly in your body when you get the common cold. So the virus enters the cell. So as you were just saying, it's like a lock and key mechanism. So these are the keys and these are the locks. And then the virus will fit in and come inside the cell, where it's taken by the sorting proteins. So this is the coat of proteins which covers the virus and sorts it to the right place. So then it takes it and then it releases the coat, and then it will, so what this virus has to do now is it releases its genetic information and sends it to the nucleus of our cell. So what happens is this genetic information is then sent to the nucleus, where it is transcribed, which is so what basically means is this information is read in the cell and no new proteins are made out of it. So this is the information being read, and this makes RNA, which is then sent outside of the cell. So as you can see, it makes multiple copies of the same RNA again and again. And this is a translation, this is a transcription machinery, which is called as RNA. And yeah, and then these are sent to then these are sent outside the nucleus, and they are and these proteins are so these proteins are then packaged into the viral particle, and then those are emitted out of the cell. So this is basically how a virus infects your cell. So as you can see, if this one cell gets infected, then it so basically what happens is one cell gets infected, the viral particle enters that cell, it causes it to make more duplicates of itself, and then it bursts open the cell, and then all the virus particles come out. So that depends on the virus. So it's very virus specific. So for example, if you have something like common cold, it will not take a very long time, because common colds generally last for 2 to 3 days. So you will fall sick, the virus enters your cell, you get a bit of a trouble, and then in 2 or 3 days it gets resolved. So, but for example, for something like dengue, it takes a longer time. So that is what the incubation period of the disease depends upon, as to how long the virus takes to cause a symptom. So what happens to the infected? So in specifically in case of coronavirus, what happens to the infected people? So what happens is, so this is our normal, so alveoli is a structure in the lungs which is used to exchange gases. So this is how we breathe and respire. So in a normal alveoli, it is open and gaseous exchange can happen. So the carbon dioxide emitted by your body is removed, and fresh oxygen is taken in. So in the lungs of a person infected with coronavirus, what happens is something called pneumonia, where this gaseous exchange is perturbed. This gaseous exchange cannot take place because the alveoli is blocked. So this is how it shows up on an X-ray. So on an X-ray, if you can see this region, so it's a slightly whitish sort of region. So that is how it shows up on an X-ray. That the whitish part is called the ground glass opacity, which is found in lungs of infected people with not just coronaviruses, with all sorts of pneumonia. So how do we fight back against viruses? So the one way to do this is by using vaccines. So we'll see a short clip on how vaccines work. So vaccines can be made from either the live virus the attenuated version of the virus which is not harmful, a part of the virus, a man-made copy of the virus where there is a man-made copy, or a virus which is not able to cause the infection. So that is given to a person, and this virus then causes antibodies to form in the body, and these antibodies fight against the germs, fight against the viruses, and then they protect you from disease. So this is something very recent research. So it has just come up, and I just drew this by hand, so I am sorry if it is not very nice. But basically what some researchers have done is that they have produced this recombinant ACE2. So I told you that coronavirus binds to ACE2 in our cells and then enters the cell. So what they have done is they have created this recombinant ACE2 which can bind to the S protein, and thus it does not allow the coronavirus particles to enter the cell. So this can be a possible therapeutic against coronavirus. So this can be possibly used to to protect people who have coronavirus and give them medication so as to make them recover. So now we've talked a lot about how the virus has been and how does the virus affect you and how does the virus move and etc., etc., etc. But what has the most viral thing about this in this epidemic has not been the virus, but the misinformation that has been going on. So this video that was this video that was shown in the internet, then this chant that was to contain the coronavirus threat, this boiler chicken mein coronavirus, all of this has been denounced as fake. So please keep yourselves aware that all of these things like these are fake, and they don't. Sorry. No, so it's not because they are stronger, it's just a matter of how much, so it's a matter of viral dose. So how much virus did you get when you were first infected? So that could be any number, like you can get 10 virus particles or you can get 10,000 virus particles. Now if it was just 10 virus particles, then maybe your body could fight it off, so you don't get infected. But if it was 10,000 virus particles, then your body couldn't fight it off, so then you would get infected. So it depends on that, and also on the health of the person. So if you have something like a pre-existing sickness, like if you have say a cold or something beforehand, then you have greater chance of having another infection.
Basically, the children are less affected than adults.
So it is known that older people are more affected. So this was also the case with the previous coronavirus, SARS, that older people were more affected, and older people, so the fatality rate was higher in older people. But it's like as of now there are no numbers, exact numbers to say that, but it seems like that is the case, that younger people are less affected, and even if they are affected, they are less prone to dying. So now what has been done is Facebook is removing these posts so that the misinformation does not spread, and myths are being debunked with help from WHO. And even the Ayush Ministry released this, but then again this was withdrawn. So please keep yourselves abreast of all these things. Don't believe everything that comes on the internet to be gospel truth. It is not. It might not be. Keep a calm eye and look at what is going on, and then make a decision as to whether it is right or not. So how scared should you be? So this is just some numbers to show you.
If somebody asks you a question, just repeat. People can't hear your question.
Achha. Okay. So yeah. So from October to February 2020, about 30,000 people have died from flu, just normal simple plain flu in America alone. So the numbers that so somebody was saying that 1,000 people have died from SARS, from coronavirus. It's not, the number is not very high, because you can see even from something as common as the flu, people do die on an average daily scale. So So just on hearing the numbers that 1,000 people have died, please don't get scared because of that. 1,000 people have died, but 60,000 were affected, out of which 1,000 died. So that's a very important part to remember.
Why the panicking more right now? Why are they panicking now?
So that is what we should not trouble for ourselves. So because we have to remember that if 1,000 people have died, 60,000 were first affected, out of which 1,000 died. So it's not necessary to say that if you get coronavirus, there is a high chance of you dying or something. So if you are a normal, healthy person, then it's a very low chance that you will actually suffer anything serious from it. So that is, I mean that, yeah, yeah. So the question was that is there a possibility that this number, this 1,000 figure is under represented? Is that was the question, right? Yeah. So I mean there is a possibility because it's China and we don't really know, but I mean we don't really know actually. So we can just right now we can just report the numbers that they have given us. So nobody can really tell. But I mean I really don't think they should do something like this because that would be really callous. But I don't really know if. Yes, yes. So the question is the panic because it is spreading so fast, and and it is true to an extent because so the virus is very infective, so it can infect a lot of people very quickly. And there is something, I didn't put it in the slides because it was too into the details, but there is something called super spreaders. So say for example, if I don't, but if I had the coronavirus and I am talking and I am talking in front of all these people and I am releasing respiratory droplets with the virus, then I can infect all of you, right? So there would be some people like that who are called super spreaders who can spread the infection to a large number of people. So in every epidemic there are some cases of super spreaders which can spread the virus to a large number of people and cause the infection to spread faster. But so we are panicking because it's spreading very fast, but there is no real reason to panic because it's not going to. So if I get the coronavirus, for example, there's the very high possibility that I will not die because I am a healthy young adult. I will not die. So there's no need to panic as such. So what are the takeaways for today? So viruses and humans have been around forever. So we have been dealing with viruses for a very long time. It has been, we have all sorts of viruses around us from flu to everything, and we've dealt with them very nicely. So there is no reason why this coronavirus should be a aberrant virus which will cause more deaths or anything, etc. However, we should be cautious. We should be cautious. It's not that we should not be cautious. We should take general preventive measures like hand washing and controlled sneezing and hygienic sneezing, etc. And no need of a panic reaction. You don't need to panic because you are not going to die from coronavirus. Very likely, if you are an adult, healthy person, you are not going to die. Older people are at greater risk, so please be careful. Exactly. So for healthy individuals, it is not at all life-threatening, and most deaths have been among the older or diseased patients. So if you are older than 60, 65, or if you have some other disease which is called comorbidity, which is a disease which is present along with the coronavirus disease. So if you have, say for example, diabetes, or if you have, say for example, asthma, then you are at greater risk. So please be careful if you have some of those, and stay away from rumors. They are more deadly than the virus. Because this virus is not at all going to kill you, but the rumors might just kill you. And this is the current COVID status. So this is a site which shows exactly what numbers the disease is at right now. And so right now, just before we started this presentation, the number was 69,256, and out of which 1,669 have died. So this is a site that you can use to live track where the virus incidences are. So you can see the maximum cases are here, but if you go a little bit to this side, you will see that India also has a couple of cases of coronavirus now. That's it.
Why are so many travel advices? Why are towns in China being not allowed to travel outside? We are not. It is not that fatal.
So the question is why are there travel advisories being issued if it's not so fatal? So the reason is we don't want this virus to spread. Just because it's not fatal to most people doesn't mean you should allow it to spread, right? You have to control the spread of the virus somehow. So it's easier to issue travel restrictions so that so there are certain, as I said, the older people who are more at risk. So if you don't issue travel advisories, those can be affected. So there is a certain population of immunocompromised patients, like patients who are getting treated for cancer and stuff. Their immune system is compromised, so they cannot fight against this virus. So even if it's not deadly for us, it may be deadly for them. So as to protect them, these travel advisories and everything has been issued.
Why do you say that older people are more affected by this virus?
So older people are more affected in the sense that, the immune system of an older person is a little weaker than the immune system of a younger person. So for example, if I compare between us, my immune system will be more stronger, so it will be more able to fight off diseases better. So even if I do get symptoms, those symptoms will not be that aggravated. But if in your case you get it and you have those symptoms, it might be a little more aggravated, so it might be a little more deadly.
But why is it something to do with the age?
Yes, it's something to do with the age. So as we age, our immune system gets weaker and weaker. So all the systems of our body get generally weaker, and so does the immune system. So you are not as able to fight viruses or other kind of pathogens.
Anusha, there is a question from the Childwise Facebook page. There is a gentleman called Suri Cornelio, and he is asking why is it difficult to attain permanent immunity against the common cold?
So that's a very good question. So the reason why you can't get permanent immunity from common cold, just like you get from chickenpox. So if you get chickenpox once, you don't get it again. But why can't you get immunity from common cold? And that's because there are lots of viruses which cause common cold. So I showed you in the viral diversity slide that there were 3 types of viruses: adenoviruses, rhinoviruses and coronaviruses, which cause common cold. So there are different types of viruses, and each of these viruses have different strains which cause different common colds. So then you have all these different strains, and you need to develop immunity against each and every strain to be able to protect yourself from the common cold. So now if you have, say, a strain A 1 month ago, then you will not be protected from strain B after 2 months. So you won't. So you will get the common cold again, and then again so forth for different strains. Anything else? What makes a virus jump species? Very good question. So the question is, what makes a virus jump species? So how it happens is basically, so I told you that viruses can undergo changes in their genetic information, which is called as mutation. So when there is a random chance that if, say, example, there is a virus which is present in, say, bats, it can affect humans. So what happened is, this virus somehow entered the bloodstream of a human, somehow by eating the bat or by a bite from the bat. It could be from anything. It enters the bloodstream of this human. Now in this human, it undergoes a mutation or a change which makes it cause a disease in the humans, and from there it can mutate such that it can transfer from one human to another. So that is how these zoonotic diseases spread. They first go from animal to human, and then they become human to human transmission diseases.
Is it primarily when they eat meat, is it because it is not cooked completely, or
Sorry?
is it because it's not cooked completely, or is it because they're eating raw, or can some of the viruses get transmitted when the food is cooked?
So generally, so the question is, can these viruses be transferred only when the food is raw, or can it transfer even when it's cooked? So generally, viruses don't survive heat. So if you cook your food, you should be fine. So generally, if you're not in contact with raw meat, you should be fine from zoonotic diseases. Sorry?
Can you speak a little louder, ma'am?
Yes. Yeah. So why is it so? So the question is, why does chickenpox have not so many strains, whereas common cold has many strains? So it comes to the point where I told you about how viruses can have either DNA as their genetic material or RNA as their genetic material. So if a virus is a DNA virus, it does not mutate that faster because the chemical property of DNA is such that it is more stable than RNA. So it does not mutate faster. So DNA viruses don't have that many strains, whereas RNA viruses change very often, so they have a lot of strains. No, so viruses can be double-stranded DNA or single-stranded DNA or double-stranded RNA or single-stranded RNA. It's the chemical difference between DNA and RNA which makes it so.
So the chai part has arrived on the chai and wine. Let's have chai and then come back again. Online audience, kindly note that we are breaking for a cup of tea. We shall be back in 7 to 8 minutes.
Sorry, can you speak a little louder?
Laundry list of all the animals which could have spread this disease, right?
Yeah.
So since we don't even know what is really responsible for that, so other than the preventive steps that we can take to contain this, at a very basic level, is it not important for us to also look at what could be the cure for, identify exactly what it is and sort of develop a cure for it?
So the question is, so because we don't know from which animal this virus spilled over to humans, how can we prevent against it?
So one is that we are already taking some measures.
Yeah.
You're controlling your travel,
Yeah, exactly.
and all that, which is happening at a very different level. But I'm saying if you don't even, at a very basic level, if we're not able to zero in on what really cost it and actually kill that, is this going to really help?
So we don't really need to know where this virus came from as long as we know what it does in our system. And we know that now, that it binds to ACE2 and it enters our cells through ACE2. So if we have to target or make drugs against it, we can make drugs against something that affects, just say I showed you the antiviral therapy which has recombinant ACE2, so then it can't enter your cells. So we can develop preventive measures like those. We don't really need to zero on which animal this disease came from, but yeah, it would be good to know so that we can be prepared for the next time. I mean, if we learn from our previous mistakes, like we did not learn from bats and civets and SARS, we did not learn from that. So hopefully next time we will learn if we get to know.
We reading that the virus spreads faster than colder temperatures. So can we assume that something like this will not spread very fast in a tropical country like India?
No, so that's not something that you can. That's not something that you can think of. I don't really think that that's correct, because especially in a tropical country like India with the overpopulation that we have, if it spreads here, it will be very rapid. But there is no need to panic. There is no need to worry. Even if it does spread, most of us are going to be very healthy. Why haven't we invented the vaccine of HIV yet? Great question. So the reason we have not invented vaccines for HIV, so we have some sort of therapy for HIV. There are two drugs which are used together and they are used to fight HIV. But we have not been able to develop a vaccine against HIV is because the HIV protein changes a lot. So when it is in your system, it changes a lot. So as I said, there are changes in the genetic information of the virus. Those changes are very rapid in RNA viruses. So it's very hard to make a vaccine against them that will be able to stay there and kill the viruses for every change that keeps on occurring. So that is why we've not been able to create a vaccine for HIV. So as far as I remember, smallpox is at least a DNA virus. I don't remember about polio, but I remember that smallpox is DNA. So it was easier to create a vaccine against that. And also, it was easier to create a vaccine against smallpox because we had this related disease called cowpox, which does not infect humans, but it is a very close relative of the smallpox virus. So it could be given to humans without causing a disease, but it could cause the immune response such that the antibodies could be produced. So that is why it could be used as a vaccine.
I want you to repeat one of your slides, whether viruses are dead or living.
Sorry?
Viruses, whether they are dead entities or living entities.
So that is a very...
He wants you to go back to this slide.
Okay. So are they dead or they living? Is that your question? So, so this is a topic of research. So as of now, we don't really have an answer to whether viruses are living or non-living. But I was reading about this the other day, and in 2008, people found viruses which can infect other viruses. So if you have a virus that can infect another virus, then that shows that viruses must be alive in some way for it to be affected by a virus. So this is a bit of a complicated thing, But as of now, we are saying that yes, viruses are alive because they can be infected with another virus. So it can, if a virus can be infected by something else, then it must be alive. It has to be alive for it to be infected, right?
In case of SARS and in coronavirus, patients who have recovered, have they shown any structure difference against further strains of coronavirus?
So they will show defense against that particular strain of coronavirus.
Currently, people not recovering as such.
They will not get this particular strain. But if there is a different strain, then they might get it.
Generally saying that for common cold and cough, you should generally not take any medicine.
Hmm, hmm.
You automatically
Hmm, hmm.
creates resistance. Is that true?
Yes, so for common cold and cough, and you don't need to take any medicine. So generally what we do in India is that whenever we get a cold, we just take some antibiotics. That's not the right thing to do at all, because some colds are caused by bacteria and can be treated with antibiotics, but some colds are viral and they cannot be treated by antibiotics. So if we take antibiotics, in that case there is no benefit to that. So what we should do in such a case is just take medicines which relieve symptoms. That is called symptomatic treatment, where you treat the symptoms. Like if you have a cold, you take something like a Benadryl, which helps just with the cough and the cold. If you have something like a fever, you take something like a Crocin, which will help you reduce the fever. So you take those medicines, but you don't take antibiotics. So so if you want to take antibiotics, you should wait. You should see whether this is a viral infection or a bacterial infection. So viral infections generally resolve themselves within 2 to 3 days. So if you go to the doctor the first time and you tell them that I have a cold, they will not give you any specific antibiotics. They will wait for 2 to 3 days. If you don't recover by then, then that means that you have a bacterial infection, then they will prescribe antibiotics.
So, how do you know when the infectious period is over, and what does that mean, and when will it stop becoming?
So that depends from virus to virus. So for example, for a common cold virus, you are infectious from the moment you intake that virus to the moment you show your symptoms. So till the moment you cough, till the moment you sneeze, you are spreading virus particles out. So you are infectious till then. For example, for in case of HIV, you are infectious for eternity, because as long as you live, you are infectious, because that virus is in your blood and it will circulate.
So, I'm saying with the common cold, how do you know when you stop becoming infectious?
So when you stop sneezing.
Why aren't there antiviral medicines just like there are antibiotics for it?
That's a very good question, and that goes into how viral biology works. So the thing is, viruses are not like bacteria. So bacteria are very similar to our cells. So it's very easy to develop stuff against them that can kill bacteria. But the thing is, viruses are not like that at all. Viruses are just some other breed of animals. So they are very different. So the thing is that they cannot survive without a cell, right? As I said, that they are not alive until they are infecting a cell. So if you have to target them, then you have to target some part of your own cell, and you have to make it specific such that it affects only that virus infected cell and not your own cells. So that's why it's a little hard to create antiviral therapies. Any other questions?
Tell us something about the viruses like mumps, which remain dormant, will get reactivated after many years.
So I don't know a lot about that, because but what I do know about that is that what those viruses do is that they form. So in I think in case of mumps, but I'm not sure. In one particular case, what they do is they form virus propagules, which what they do is basically they stop their phase. So when I told you guys that when a virus infects a cell, it causes the cell to burst open and then release more virus particles. So what these viruses can do is that they can control this process. They can cause the cell to not burst open, but keep on making new virus copies and keep on secreting those virus copies slowly by slowly. So then what happens, this is called lysogeny, where the virus is not actively killing your cells, but it is creating more copies of itself. And when it transfers from a lysogenic to a lytic state, then it will kill your cells and release all your virus particles.
So we have a question online over here by Jay Satmani. Are any natural foods like turmeric, cinnamon, ginger, garlic, etc. useful in fighting viral infections?
So so the question was, are natural elements like turmeric, ginger, etc. useful in fighting viral infections or no? So generally generally turmeric is known to be an antibiotic, not an antiviral, so you can't really take it for viral diseases. However, what these also do, the turmeric, ginger, etc., what they also do are they are symptomatic treaters. Like if you have a cough and you take ginger, it soothes your throat. So that is why it helps. So it will not help in case of fighting a virus per se, but it will relieve your symptoms. So it's no harm to take ginger if your throat is hurting. Basically, what's the question? Oh,
So another question is from Suri Cordelio. Can we use bacteriophages against bacteria rather than using antibiotics, as they tend to have some side effects?
We can use bacteriophages against bacteria, that's a very good idea, but we need to make sure that those viruses don't infect us. So we have to be very sure about the fact that those viruses won't mutate into something that will cause a disease for us. So that's why it's not a very good idea to do that, but yeah, I mean it can be taken upon by someone, research can be done upon that.
Next question is from Amit Nirani. Any idea about size of N coronavirus? Whether use of mask actually prevents coronavirus transmission?
So yes, I'll like to explain something here. So what happens when you have droplet transmission, as I talked about it, so you sneeze or you cough out droplets of water in which the virus particles are there. So you are not trying to prevent so the mask is not trying to prevent the viral particle from getting inside. It is preventing the respiratory droplet from getting inside. So the respiratory droplet is of a huge size compared to the virus. So if a virus is, let's say, 75 nanometers, the respiratory droplet is at least 1,000 times bigger than that. So the mask will prevent the transfer of the respiratory droplets from the air to your mouth. So it's not going to prevent each virus particle individually, but it will protect against the respiratory droplet.
Isn't it a fact that people who are infected with this kind of viruses, they should be wearing the mask
Yes.
rather than the general public
Yes,
going
yes,
out in
yes.
medical stores and flooding and buying those by dozens?
Yes, exactly. So if you have a cough or a cold, then you wear a mask, then it makes more sense, because then you are protecting everybody else around you. If you don't have any symptom, and you wear a mask, you are just protecting yourself from any sort of virus that might be, any sort of pathogen that might be in the air. You are not really protecting others from it. So it's not really that useful. What is useful is that persons who are infected with something, who are sneezing, who are coughing, they need to wear masks so that they don't spread the infection to others.
Are there any antivirals against coronavirus? If yes, then how does it act against the virus?
So no, as of now, so coronaviruses have been around for a very long time. So if you're talking about this specific novel coronavirus, then there's no antiviral therapy as for it as of yet. For SARS, there have been some in discovery, so process is still going on. So the process of making an antiviral is a very long process. So it takes a lot of treatment and a lot of research, and a lot of things go into it. So you can't make it very fast. So that's why it's not really there per se at the moment. But there are therapies which can be used against it. Like it's an idea that, okay, yeah, it's a cool idea, we can probably use it to fight infections. Let's try it, but not a cure per se, and especially not for this novel coronavirus.
So if we use a normal handkerchief as compared to a mask, is that better or bad?
Sorry?
Instead of a mask, actually a lot of people use handkerchief.
Yeah, So handkerchiefs, so the thing is, masks are actually made to protect against bacteria because bacteria are bigger. So masks will protect, so the point of a mask is that it will protect against bacteria as well as viruses. Your handkerchief will not protect you against bacteria, but it will protect you against viruses because it will not allow the respiratory droplet to go out. There was a question. Sorry? So the question is, can animals get affected and then can they pass the infection to humans? So in a lot of diseases this does happen, that animals do get affected, and so the thing is, these are all probability numbers. Whether is there a probability of this virus being transmitted to an animal, then there is a certain probability that yes it will, or no it might not. If then that happens, then there is a other added probability of whether it can come back into a human or not, yes or no. So there are two possibilities. So what you basically do is you multiply these probabilities together, and then you figure out the final probability of this event happening, this virus from going from animal to human again. Sorry? So relapse. So if you've had coronavirus specifically, then you say, for example, I talked about this particular strain. Yeah. So for example, if I talked about this particular strain, so we've all had this, most likely we've all had this. So if we get this exact strain of virus again, we will not fall sick. However, if we get a slightly mutated version of the same strain, then yeah, we will be affected. So it depends on whether what strain you are catching again. If you are catching the same strain again, then you won't be affected. But if you are catching a different strain, then you will be. So I don't know how these are numbered, but what I do know is that there is this body of scientists called something virology nomenclature something, which decides which virus will be named what. And so it's basically, if I think it would be just how the virus was detected and how it was, how the strain was isolated. So the numbers will be based on that most likely. No, so I did not put in here for more because I did not want to put more information. But there are also there's alpha, beta, there's delta, and there's gamma coronaviruses. So there are 4 types of coronaviruses. Yeah, here I have put 2 alpha and 2 beta. There are also 2 delta and 2 gamma. They are not very common, so they don't generally occur in the people. That's a very good question. So the question is there a common evolutionary history for viruses? Where did they come from originally? So let me think about that. So yeah, I mean, obviously, like everything has an evolutionary past, viruses will also obviously have an evolutionary past, and we can see, so in viruses evolution is very fast. So for us evolution means in a time scale of millions of years. For viruses that means maybe days. Because they mutate so fast, it can be a matter of days. It depends on the virus, but mostly it is a matter of days to months. So it's very hard to track viral histories like that. And I have a hidden slide which I can show you. Yeah. So this is how you trace out evolutionary history of viruses. So what you do is you go back to the original source, you check how much times the virus has changed, how many times the virus has changed, what changes have happened, and you take a look at that and you make something called a phylogenetic tree out of it, and then you see, okay, this virus has changed in like say 20 places, the genetic information has changed in 20 places. So this must be a new version. However, if there is something which has happened for 50 changes, there is 50 changes which has happened. So that means that must be even newer than the 20 changes. So that is how you calculate, back calculate ages of viruses.
So, assuming that mutations are random,
Hmm.
if a mutation is going to give a virus a better chance of survival, it could also happen the other way that it could.
Yeah, sure.
A lot of viruses.
Sure, of course. So the question is that if a mutation can give the virus a chance to infect humans or infect more people, then a mutation can also give them the chance to make them less infective and less virulent. So that is obviously true, and there are cases of that. It's just that we don't know about it because we don't care. We don't want to look at that, right? We don't care about that because if a virus gets less deadly, then it's not really much for our, it's good for us. So we don't really study that. Yes. So yeah, exactly. But the thing is that the rate of mutations is so high. That the percentage of so if let's say for example they have 100 mutations, out of which 20 make the virus more infective. So then we get to know about those 20 much more than we will get to know about those 80 mutations that cause the virus some harm.
If the mutation takes place faster, because finally the aim of each cell is to multiply and divide, or divide and multiply. So does it get faster or does it get slower down because of any reason, or the viruses make them slower or faster?
Can you repeat the question?
What I'm saying is that the mutation is basically again multiplication and division of the cell. So if the rate of division and multiplication gets enhanced by the virus, or it will get reduced by the virus, or does it remain the same and the process takes place naturally by itself?
So if you are referring to viral replication, so the replication of the viral particles to create more viruses, are you referring to that?
That is natural. But is the process get faster or retarded because of the virus present in the system, or it is
No, so a virus will not affect what happens to your cell. What a virus affects is the replication of its own copies. So what the virus will do is it will make your cell make more copies of itself. But your cell will not have any changes in its replication rate or anything. I mean, there are some viruses which can cause cancer, and in that cases the replication rate of your cells will be increased, but that's a very rare scenario. Okay, so that's a very good question. The question is what changes in our body such that it makes us less susceptible to a second attack. So and the way it works is the first time a pathogen enters your body, you have something called immune cells which take care of it. So what they do is they create antibodies to fight against it. What they do is those antibodies cluster around the viral particle and they don't allow it to enter another cell or do anything else which will harm you. So those antibodies are made in your body. Now those antibodies are stored. So there is a certain population of cells called memory T cells. Those memory T cells stay in your body for a very long time. So if you have had an infection like 2 months ago, those memory T cells will still be there and they will be there for a very long time. So the next time you get infected by the same strain of the virus, then those antibodies will act against that immediately and you won't have to go through the whole process of your body generating new antibodies and then making copies of it and then and then troubling the virus, and you won't have to go through it again. You will just have ready-made antibodies against it. So they have a lifespan, but it depends from virus to virus also. So for example, for chicken pox, if you get the virus once, your memory T cells form such that they don't die. So they are there for a very long time. So then you don't get affected again. But for something like common cold, the memory T cells might not live that long. So then you might get it again.
What is the simplest example of an immune system present in that?
The simplest example. So the simplest example is a bacteria, is a system in bacteria, and which is what we use for. So I guess some of you must have heard about CRISPR. Yeah. So CRISPR is a process by which you cause gene editing. So that is actually something which was present in viruses as a form of immune protection against that was present in bacteria to protect against viruses. So what it used to do was it used to cleave up viral DNA when it entered the bacterial cell. It used to cut up the chop up the viral DNA. So that's a simple example of an immune system.
So these air of this droplets of water that one might cut out during an infection, how long will it be talking for, or how long will it be infectious?
So that's a very good question. So that depends from a different virus to different virus. So for measles, so as of now it's not yet known as to whether, so if I sneeze say on a, if I sneeze and then touch my phone, will my phone have viral particles that can infect you like 2 days later? At this point we don't know that. It could be, so it's still better to take that precaution, but right now we don't know. So in case of measles, for example, if you sneeze, say if I sneeze in this room and I have measles, that virus particle can stay alive for not stay alive, but they can remain dormant in the air for 18 hours and they can infect somebody 18 hours later and cause the disease. So that depends on virus to virus.
All viruses may not be bad. Are there any beneficial viruses for humans?
So I will create a lot of controversy if I say this, but HIV is actually very beneficial to humans. So very beneficial to humans. So we use it in research a lot. So this I showed you, this that I showed you, this was done using HIV viruses. So what they did was they used HIV virus ka body and they coated it with the S protein of coronavirus and then they used it to study. So HIV, the virus HIV replicates very fast and it's very good to easily work through it. So that's why what they did was they just kept the S protein of coronavirus, but they kept the viral body as actually HIV, and then they used it for research. So viruses can be used, are very easily used in research. There are cancer drugs that are based on viral particles. So there are these viruses called lentiviruses, which are a major part of cancer therapy. So we do use viruses a lot. Sorry. So what can be done is that this coat is, so I showed you that video where the viral particle was assembling. So you first have the DNA or the genetic material, then you have a coat of proteins, the coat which forms, then you have proteins on the top of that coat. So these proteins are modular, like they can be removed, like अगर यहाँ पर से एक हटा दिए and दूसरा लगा दिए तो it can still work. So that is why even you have the body of an HIV virus, you can still have the coat proteins of coronavirus.
The cancer can also be a virus, basically cancer is also like a virus?
Not really, and there are some viruses which can cause cancer. So there is this one particular virus that I remember, human papilloma virus, which can cause cancers, but generally they are two different things.
So there is again also a mutation which is.
So that is something that happens in your own cells. So virus is an external agent which comes inside and then mutates in your body. A cancer is when a cell inside your own body mutates and causes changes. Yeah, they must have recovered. Yeah, so if I go back to this, I think, yeah, so they have this. So they have this number, total recovered. So total people who have recovered in the sense that their body does not have any sign of the virus particle now. So that number is given here. But and the rest, so 1000 have died. 9,000 have completely recovered, but the rest of them still have some amount of virus still left in their body. Any other question?
So we have one more question here by Surit Cornelio. The N95 mask can prevent droplet contact, but can the virus still infect us if it makes contact with us?
Yeah, so that is why, a virus can infect you if it comes in contact with your system. So what you have to do is, you have to make sure you wash your hands. That's why washing hands is very important. If you wash your hands quickly, then if you're, say for example, if you're not washing your hands and then you're eating with those hands, then those viral particles will get transferred into your body. So you should wash your hands before, and then just use the mask to create a barrier, and then wash your hands if you come in contact with something like a virus. Sorry? Yes. So the question is, can you get asymptomatic infections, where you get the infection but you don't show any symptoms, and that happens in a lot of viral cases. So for coronaviruses, it's not yet known. For this novel coronavirus, it's not yet known whether that can happen or not. But for SARS, it was known that generally if you had infection, you should show symptoms. So right now it is speculated that because it is so similar to SARS, it must also work the same way, but it's not known right now. But generally, yeah, there are certain cases where you can have the viral infection but not show any symptoms. If your immune system is strong enough to handle it, then you won't show any symptoms.
But you will still be infectious.
Yeah, you will still be infectious. You will still be infectious.
In the case of tracking viruses and find where they came from.
Sorry?
If you're trying to find where the virus originated from, how do you generally find the patient zero?
So I don't know this about coronavirus because I did not read that much about because it's still the epidemic is still under progress, so there's not much clear information about who would be the originator of this virus. But I know this for HIV, and this is a very interesting story, so I'm going to tell it. So what happened in HIV, what is speculated that what happened in HIV was that there was this in Africa, in Congo, there was this man, a Bantu hunter, a Bantu tribe person, who used to hunt chimpanzees. Very likely what has happened, that chimpanzee was killed by this man, and in the process of that hunting, this man was also cut somewhere. So this man had a cut somewhere, and the chimpanzee blood somehow entered the wound of this man and got transferred into a human. So this virus, the human immunodeficiency virus, has a relative called the simian immunodeficiency virus, which is found in chimpanzees. So what is said is that SIV got transferred from the chimpanzee blood to the human blood, and from there it originated. So that hunter then must have traveled somewhere, and there he must have done something with other people, and so from there the HIV infection spread. So you can track patient zero like that, but honestly it's very hard to do that. It's all speculative. I mean, you don't really know who was the person. There is a record of the earliest record of HIV is from 1920, I think. So from then, so that is the first time HIV sample was recorded from a human being, and so that tells us that HIV had transferred into humans from 1920. But what we don't know is how that process happened. We can only speculate कि हाँ ऐसे हुआ होगा.
If there are no more questions, let us thank Anusha. So I hope any of you will join us at the EIFR to enjoy the National Science Day, which we are celebrating 5 days in advance, because our team will be traveling to GMRT Kodak for the actual mountain day. Have a nice weekend.
मेरा पूरा बायो का रिवीजन हो गया यार.